Okay, so here's the plan for today and next time. Slight remix of my plan, but not too much. So for next time, we got a paper discussion lined up, and I found the paper. This is it. I'll send it by email, but this is something I just came across recently. It was just published in 2024, and it's on my list of papers to read. So it seemed like a good opportunity to do that. And probably a lot of people are interested in some of these issues here. So that's what we're going to do next time is, we'll spend most part of the time discussing this paper. But then, maybe to have make it easier to understand this paper next time, there's some like preliminary info that I think y'all should have. So I found basically two things in here that we haven't talked about yet, which are temperature and variable quotas. So I thought I'd give you all a little introduction to those topics. And then next time you'll be prepared for that. And then we also have spend some time discussing this thing and its hangers on. And my original plan was to do that first, discuss the papers kind of building on last week and then get into the new lecture part. But then I was starting to be worried because it's I don't want to go over and I don't want to have 20 minutes blank at the end. So I think the way to do it is start with the kind of lecture-ish material and do that. And then when we're done, we can use the paper discussion part to fill up the time adequately. So that's the scheme. One other topic here is next week is spring break at Michigan State, but the majority of people here aren't at Michigan State. So I think we should just go on and do it next week as usual. And people who are enrolled for credit MSU, you're not obligated to come, but you're welcome to come. Just because we lost, a few weeks at the beginning of the semester and there's so much resource competition theory out there. I don't want to waste another week just when we're getting some momentum here. So that's what I got for introductory thoughts. Any comments, questions on that? All right, so let's do this. So yeah, so basically I guess we can cover, we could put all these things we're going to talk about to warm up for next week's class under the umbrella of more environmental and physiological realism. And we'll cover sort of three things here: environmental factors, temperature in particular is a particular environmental factor. And then we're going to switch gears and talk about this variable nutrient quota. So, all right, environmental factors. And as we discussed, was it last time? An environmental factor is not a resource. An environmental factor is something that affects performance, fitness, growth, but it's not consumed or impacted by the populations. So it's kind of like a one direction, causality here. So you don't have that sort of feedback loop that you have with a resource where resource spurs growth and growth, depletes the resource and therefore it sets up some sort of negative feedback loop that leads to density regulation and prevents things from blowing up to infinity. So environmental factors just one directional. They affect the population, but not vice versa. And so how can we plot the effect of an environmental factor? There's lots of different ways we could do it. Here's a classic one, the Gaussian. This goes back at least to Gauch and Whitaker, who had, a kind of cool early paper, but they cite earlier papers than that. So it's kind of like deep rabbit hole here. I should say Gauch and Whitaker weren't so much talking about how the environment affects performance per se. They were looking at how the environment correlates with abundance along an environmental gradient. So slightly different. But anyway, I thought we would just hijack it anyway here. So with the Gaussian, it's defined by three parameters here. So we're plotting fitness or maximum growth rate somehow as a function of this environment. And so there's three numbers. There's the optimum environment where you reach your maximum growth rate. There's this mu max, which is your growth rate in the optimum environment, let's say. And then there's the width of this Gaussian function, kind of like a specialist generalist kind of thing. Okay. So let's start off with this. And imagine that there's a trade-off now between two species with different environmental optima. We want to understand competition between these species along this environmental gradient. Well, we already have the machinery to do that. Maybe it's as simple as whoever's got the highest growth rate wins. But based on resource competition theory, and that'll actually turn out to be true here, but it's not generally true. What is generally true is if there's competition for a single limiting resource, then whoever wins competition is the one with the lowest R star for that nutrient. So instead of plotting the growth rate, we can use that, plug it into our formula for R star as a function of the environment, and then plot that. So instead of those Gaussians, now we get these kind of like, unimodal, anti-unimodal, U-shaped curves. R star is a function of the environment. And what can we see here? So first of all, well, with the Gaussian, this won't quite go asymptotic, but there's a strong, like, this is kind of getting at the range limit of like the fundamental niche of this species, because if your R star is infinite, then you can't live. So species 1 could live anywhere in this U-shaped region. Species 2 could live on its own in its U-shaped region. But where they overlap, that's where we have potential competition. And so for a given environment, the species with the lowest R star wins. So we can use this then to map out species distribution along this environmental gradient with a picture like this. So we look for basically the intersection point where their R stars are equal. For any E less than that, species 1 is better, so species 1 wins. For any E above this intersection, then species 2 is a better competitor. So at least according to this model, if this is one resource plus an environmental factor, we can shift the identity of who's the better competitor, but we can't get stable coexistence, right? We can actually understand this in the same way as we did with the two resource zingy graphs. In fact, these U-shaped curves are kind of like zingies, zero net growth isocyclines for each species. And you can imagine a supply point being a point with the state of the environment, which is fixed, and the nutrient level. Here the consumption or impact vectors are pointed both straight down, because both species, as they grow, they consume the resource, but they don't do anything to the environment because it's an environmental factor. And then we bonk into this axis here. So here at the coexistence point, I plotted the two impact vectors, the two species, but they're the same. They point straight down. And therefore, where we would usually expect to have like a coexistence region, that little wedge that we saw last time, there is none because it's just not open because they have the exact same impact vectors. So it's just like, putting this in the context of the zero net growth isocyclines and consumption vectors and stuff we looked at last time. Any questions about this so far? Pretty straight.
I have a question.
Oh yeah, go on.
Oh, sorry, Mathilde just put her hand up, so I'll let her go first.
No,
Okay.
go ahead.
Oh, okay. So the point itself is a coexistence point, but like what's the region itself? Is it like a founder control, like either or that part in the
Yeah,
middle?
yeah, yeah. So at that point, yeah, so this would be like a coexistence point in the sense that at this point, both species have zero net growth, so they're kind of like birth equals death, so no one's going extinct or taking over the world. But this particular kind of coexistence at this point is going to be the sort of neutral kind, right? This is just the exceptional weird situation where the two species have equal R stars and therefore they coexist in a neutral sense. But if we perturb the system just a little bit, tweak it, the environment plus or minus around here, then we get into one species winning or the other. So basically the area of this coexistence region is zero area, right? Because it's just a point. And so this kind of like highlights the fact, I mean, it's kind of like reinforces from last time that to get coexistence, stable coexistence, you need also a trade-off in the impact vectors, which then like kind of like opens up this coexistence region and sort of stabilizes the point. And if if you move the impact vectors the opposite direction so that you consume more of the factor that the other guy needs because you're a jerk, that's gonna set up the founder control kind of outcome. So this is like right at the border between, you know, founder control and stable coexistence. So it's kind of the neutral point. Or going back a couple times to put into like Peter Chesson terminology, like niche overlap is 100% here. So there is no coexistence region. So does that help out?
Yeah, it helps. It helps.
Okay. All
Thanks.
right. Yeah, Matilda?
So I didn't get whether the shape, the function of R star in the environment is specifically associated with the Gaussian shape of growth rate in function of the environment, or it's just a made-up one? Like,
Yeah, so this
mm.
this one particularly, I plotted it from that Gaussian. So this is driven by that Gaussian growth response curve. But in general, if you have a unimodal growth curve, you're gonna have this kind of U-shaped R star curve. And the details will matter if you have different functional forms or not.
So if I assume a Gaussian shape for the growth rate, I would have this U form just shifted from one side or the other of the environment, basically.
Yeah, yeah. So this is exactly corresponds to these Gaussian curves.
Hmm.
I mean, it's basically, I assumed a type 1 functional response, so it's kind of like 1 over the Gaussian. So
Ah, okay, yeah.
this is kind of like what 1 over a Gaussian looks like.
Mm-hmm. Thank
We'll
you.
look at some other shapes in a little bit. All right. So, yeah, Eduardo.
Hi, maybe you're gonna get there later in this class or when we're discussing the paper that you showed at the beginning of the class seminar. But I was wondering like the relation of if you change environmental way in this Gaussian way and then you change growth rate, like I know you're talking about resource completion theory, but I was wondering what happens with carrying capacity in the lenses of
Hmm.
that. And
Yeah.
like I don't know if you want to save this for later or
Yeah. So
use
we're gonna
these
look at some different
analogous.
shapes for sure. Carrying capacity, we could look at it, but I don't have slides on it. So, in these resource competition models, maybe I'll, try and squeak those out because it should be pretty easy. In these resource competition models, carrying capacity isn't like a function or isn't like a parameter itself, but it's kind of an emergent property of, nutrient supply, nutrient uptake, mortality, and so on. Well, basically, what it's gonna look like. I think this is true. Well, imagine we've got like a single one of these. Probably the carrying capacity is gonna be like proportional to the nutrient input concentration minus the R star. So we could just like, I could probably, if I was wanted to be dangerous, I could just flip this in PowerPoint here. But it's gonna look like, yeah, this thing like just flipped over, I would say. So, and that's kind of like in a conservation of mass reason. So if you were supplying nutrients with concentration Rn, and then, they consume those nutrients down to R star, where those nutrients go? They went into the biomass, right? And here, since the R star is so low, it's really close to zero here, they almost consume everything. So the, abundance is going to be like, the carrying capacity is going to be kind of flat at the top here. And only as you get towards the edge of your range is it going to start to fall off. And then it's going to be pretty precipitous as you crash into zero. Well, with these Gaussians, maybe you would just approach zero. So yeah, that's a good question. Yeah, I know people are interested in that question. And that was like one of the reasons why we were kind of talking trash about like the lack of Volterra model. Because parameters are kind of like, where do those come from? How do they relate to each other? So this shows it's not so simple. All right, so a little bit of, here's an example, an experimental, is this, where is this cited in these papers we're reading for today? Here's an example from Tillman back when he was still working on algae. So he did his PhD on algae and then did it for a little while, and then he switched to terrestrial plants, and he was lost. So here he did an experiment with, well, I'm not showing all the graphs. So he measured the parameters of the Minot function for these two species of diatom as a function of temperature, and calculated their R star for silicon. Put the two together, there's this predicted crossing point, and so then that predicts dominance by this Synedra over here and S. Therianella over here. So that was verified experimentally. So seems to work. Why don't people do this more often? This seems like, such an obvious thing. I don't see a lot of papers, actually testing this. But you can imagine we could extend this to more species, as showing species with a trade-off along environmental gradient. So yeah, nice theory for predicting species replacement along environmental gradients. Not so good for coexistence in the sense that you'd have two species in the same environment. But if you step back and look at the whole gradient, then it's a really good model for coexistence because everyone's got their spot and, they're hanging out there. All right, so that's all I gotta say about generic environmental gradients. Let's look at one in particular that a lot of people like to study, including us, this temperature. Temperature affects everything in ecology. So it's, 1/2 of Jim Brown's metabolic theory of ecology, along with body size, the other half. And he sort of resuscitated and repopularized and hyped up this Arrhenius function. So this is a function that relates any biological rate to the temperature. And so the Arrhenius function is this e to the minus e over kt. This e is not the environment anymore. This e is a so-called activation energy, which is, measure of how sensitive a particular chemical reaction is, or biological rate is to temperature. K is some number from physics, and T is the temperature. So it's a little bit confusing. I always get confused when I see this thing because it's got this minus and it's got this 1 over T. I have to work through what it means, but in essence, it means the hotter it is, the faster these processes happen. Sometimes, they plot graphs on a log scale, it's linear and 1/T. But those graphs really confuse me because then like you have to remember hotter is to the left. Well, that's great, but real thermal performance curves, they sort of follow the Arrhenius or kind of exponential increase at lower temperatures, but typically then they reach some optimum temperature and then they fall off. Okay, because it's too hot. That's not included in the standard metabolic theory of ecology. So I would say this is probably, I mean, it has to be, right? It has to be a universal result in ecology that thermal performance curves, or TPCs, are unimodal. Because at some point you're just like frozen, another point you're like on fire. So somewhere in between you can grow. So it has to be unimodal, right? And they're often shaped like that, and they're often kind of skewed like this, where there's kind of a increase on this left-hand part, but after you peak, you really like fall off really fast. So there's some data from Elena Lichman and Merdul Thomas, but it was just the first thing I could find in my database of some curves. They often look like this. So not Gaussian, not Arrhenius, it's something else. How to describe these? This is a classic paper by Epley, who collected growth rate versus temperature for various phytoplankton experiments. And what you see is that it's kind of, there's a lot of variation as a function of temperature, but it's bounded by this upper envelope. This upper envelope is what we can call the Epley curve. And this is slightly different than the Arrhenius. I would say a little bit easier to understand. It does basically the same thing. It's just an exponential function of temperature. But what's going on underneath here? This is the part that's like so cool. So Epley, here he's just showing all the points from all the different phytoplankton cultures. Here he's connecting the dots on different species. And now you see it's not just a bunch of points, but each species has this kind of unimodal growth curve, but those unimodal growth curves are sort of constrained by this upper envelope. Yeah, so Wu said in the chat that there's also this molecular rate theory in soil microbial dynamics. So yeah, there's a bunch of different theories out there. I should probably get familiar with all of them. There are definitely ways to get this peak. So this Epley paper inspired Jean Norberg to come up with what is now called, maybe we can call it the Norberg model. So this is published in a paper in LNO. For those of you who don't know, LNO, Limnology and Oceanography. It's kind of like the flagship journal in aquatic ecology. And so Norberg combined the Arrhenius envelope with, in his case, a quadratic function. So this is the green curve that sets the upper envelope, and then this is the part that makes any given species unimodal. And so it's kind of like inspired by Epley's findings. Oh, John himself did some modeling with this. We use this as the basis of a eco-evolutionary model, 2012, trying to understand the sort of global biogeography of optimal temperature, thermal traits. One thing that's kind of subtle about this, like we didn't notice it for like a year when we were working on it. And then when we figured it out, we're like, whoa. So we can consider for a given species the T-opt, optimal temperature for growth, but is it the optimal temperature for growth, or is it the optimal temperature where they're the best competitor? So here I'm showing one particular species, and then the Epley curve that bounds all of the species. What we see here is that what we would naively consider to be the optimal temperature for growth, that is where this particular species growth rate is maximized, it's warmer than this point over here where the TPC is touching the Epley curve envelope. And it's at that point where this curve crosses the envelope, which is where this species is like the best competitor. Everyone else is worse at this point because they're underneath the envelope. So I don't know what else to say about this except for it's a little bit subtle and surprising, and you should keep track of it. So in the model, this is like the peak of the quadratic, but this is the peak of the entire function. Yeah, Veronica?
So is this like, I don't know, maybe I'm using species distribution modeling language, but so the optimal growth is more like a fundamental niche, and then the T-op like for competitive is like the realized one? Or are
Yeah,
they
that's
essentially
probably a good way to think of it. It's certainly got like that sort of flavor in it, right? Where if the species was out there just in the world by itself, its preferred temperature would be here. But then, when you put in there with, all the other species in the world, it would dominate around this lower temperature. So this is actually like drove in our paper a lot. So we found in our paper that there is like optimal temperature for growth was a little bit cooler than the environment where they were found. This was certainly explaining part of that, this discrepancy. Yeah, Sarah?
I'm just trying to get my head around it because I had either not registered that or forgotten about it. So the fact that T-op for competition is lower than if the species is by itself, can we say it's then a signal or like a trace byproduct, how we want to pronounce, of the trade-off of resource limitation, if we consider it in the resource competition theory, if competition is for resource, being I can be best at removing the nutrient from that environment, but it means growing slower, as slower, like colder temperature is slower growth. Or is it reaching too far in the fast grower or slow grower best competitor trade-off?
I think it's related. But in this sense, this is even more fundamental than the resource competition part, because this is saying like at that temperature here, the T-op for competition, this is the fastest growing species, right? Everyone else would be like, they would be like touching the envelope over here, and then they would be underneath this blue curve, or they'd be underneath it and touching it over here. So it's not so much that like there's a trade-off between growing fast and growing slow here. Yeah, it's just that, the whole thing is kind of distorted by the fact that in general, everything grows faster when it's warmer. So anyway, it's tricky. I mean, another explanation that people would have for why you would prefer a temperature less than the peak of your growth curve is if there was environmental variation. It's like, if you go down here, it's not so bad. If you go to the right, you really fall off fast because it's so asymmetric. But this is true even without environmental variation. So anyway, I just pointed out because I saw another paper recently that was talking about this kind of, disjunct definition. Yeah, John?
Yeah. I guess two quick questions. One thing is that it's not completely trivial that all of these like species are bounded by the same curve, right? Like I understand the sort of exponential Arrhenius relationship, but couldn't you easily have like different kind of proportionality constants, like values of A?
Yeah, I think it's certainly an oversimplification to think that like, everyone goes up and, touches that curve. Here there's here this is kind of the idealized situation where the only the only thing that species differ in is their optimal temperature. But in the real world, some species, just grow slow for other reasons, right? So they would just be like kind of like down here and never even touching the curve.
So maybe did this work because these were all kind of like similar species and like similar environments besides the temperature?
Probably, but I mean, even you can see here, like, this square guy isn't touching the envelope, right? So he's probably got some other strategy going on, that makes up for his lower maximum growth rate. Yeah, it's, when you get into like multi-dimensional trade-offs, it's like it gets ugly. And plus it was 1972, and this looks like it was drawn by hand. So I think there is a little bit of like, try and make a nice picture. Yeah, Scott.
Okay, I guess I was just wondering if you think that this, not this relationship, but the one on the next slide, this one, if you think that relationship is like strong enough that you could, for example, if you had a bacteria in the lab, you could do like an empirical temperature growth rate curve, and then use this to try and like inverse model what you think the actual temperature is that they typically experience. And specifically thinking about this one bacteria, Bacillus subtilis, that is like a total lab rat. And I've always wondered why it grows so fast at 45 degrees. And this just, I guess, would explain that.
Yeah, that would be cool. We saw like some signature of this in this global phytoplankton database that Radul put together. But of course, there's a lot of other stuff going on in that global data set. So it'd be really cool to have like a nice, carefully controlled experiment showing this. But the thing is, I think that what John said, you'd also have to make sure to get species that like are, all else equal, right? Maybe different strains of the same species is the way to go, so they're not like totally different. So yeah, it'd be cool.
Thank you.
All right, so that's the Norberg model. Oh, so just one other like, like foible, one other thing that we ran into. So we wanted to take this Norberg curve and like put it into some resource competition models. So we did it the naive way, by basically making the growth function, we made it a multiplicative function of a temperature dependent growth, right? Because this is all talking about mu max, but what about like the nutrient here? So how do you model temperature nutrient interactions? The simplest thing we did was took the Norberg function and multiplied it by a Monod function on the nutrient. And that was great until we're running some simulations and weird, inexplicable results were happening. So what happened, where we went wrong, was what happens if you're outside your fundamental thermal niche? If you're in the range where your temperature dependent growth is negative, because the Norberg function can become negative because it's too hot, you're not actually growing, you're dying. The problem this caused in our simulations then was once this mu t, the temperature dependent growth term, was negative, then all of a sudden nutrients, instead of being good for you, became bad for you. So you'd have more nutrients, you'd die faster. And then this term was even worse because if you're in that range, instead of taking up nutrients, you would excrete them. And it was a mess. So we realized there was something a little bit not great about this formulation. So we changed it. We came up with this new formulation that we call a double exponential model. And I'm citing us here, but people have actually like already discovered this double exponential function before. But essentially what we have is we have two temperature dependent demographic functions. The first is the birth rate. And now it's not a function of T-opt. Actually, T-opt, we got rid of it here. It's just an exponential function of temperature. But then your mortality, your death rate, is also an exponential function of temperature. And surprisingly enough, if you take the difference of two exponential functions with different exponents, then you can get a pretty nice looking unimodal skewed curve. And so this basically the solution here is that just separating out births and deaths essentially, and making only the births nutrient dependent. So that solved our problem. It actually turned out to be kind of interesting by itself, because if you take that double exponential model, it sort of makes an interaction between temperature and nutrients. So in this bottom graph here is looking at the thermal performance curve as a function of the nutrient concentration. And you see this interaction. So like the T opt actually moves to the left under nutrient limitation. And so this graph on the right puts those together. So this is like the color here is growth rate as a function of temperature and nutrient concentration. The white line is the optimal temperature. So there's kind of like a ridge here and it moves to the right. This is the optimum temperature as a function of nutrients. And then this black U-shaped curve is that same black U-shaped curve we had before. So if we assume some sort of mortality rate, then this is the zero net growth isocline where births just equal deaths. So anyway, and then, Mridul and Elena and others have actually found empirical evidence of this interaction between nutrient limitation and optimal temperature. So that was surprising. This is just kind of like scratching the surface. I mean, of course, is still like super oversimplified, and there are lots of ways we can make this more realistic.
Hey, Chris,
Yeah.
could you explain why there's two R stars?
I think this is like just labeling the same curve at both ends.
Okay.
Yeah. So this is just the R star as a function of the temperature. Here it's at the maximum temperature, here it's at the minimum. So yeah, so if you're like outside this underneath the U, then your birth rate is less than your death rate. So all right, so yeah, I'm not saying this double exponential is the only explanation for the skewed thermal performance curves, but maybe it's one of them. That's all I want to say about temperature. We'll read more about it in the paper for next week. Maybe we'll read more about it later on too, depending on what people are into. Last thing I want to talk about here is this variable nutrient quota. So you remember in the resource competition models we've looked at so far, we've had these parameters Q, which is the nutrient content per cell, and we just assumed it's a constant. But that's good approximation for some organisms and not so good for others. So kind of part of the dogma of this, ecological stoichiometry is that maybe animals have more or less a fixed stoichiometry or nutrient content per individual, whereas others, such as plants and especially algae, are more flexible. There's a lot more variation in the nutrient content. So we want a model that can account for this flexibility. Okay, so here it is. It's what's called a variable internal stores model. And it breaks up growth into two separate steps. So first step is uptake of nutrients from the environment into the cell. They're kind of put on a pile, this pile being the quota, which is now a dynamic variable, not a parameter anymore. And then this nutrients on the pile are used to assemble new cells and growth. So V is nutrient uptake, mu is growth as before, but now they're not, fixed to each other at a constant ratio, but it depends on the internal stores of nutrients. So the way this goes, yeah, question, the type of resource sequestration, yeah, you could see that. So the external resource, just chemostat, uptake according to this V function. The equation for the quota is a little bit interesting. So the quota, nutrients per cell, increases due to nutrient uptake, and then decreases due to growth at this rate mu of Q. And we'll get into it in a second why there's this minus term in there due to growth. But essentially it's what people sometimes call dilution by growth, right? So if it's quota is nutrients per cell, and then you grow and you make a bunch more cells, and you have the same amount of nutrients, then your quota went down because you got more denominator. We'll see this again in a second. And then the growth part's pretty easy. So growth is a function of the internal nutrients Q now and mortality. This is the simple framework. This is an example of what we could call a structured population model. So a structured population model is just one where one number is not enough to adequately describe the population. You know, in this case, we have to keep track of N, the number of individuals, but also Q, the nutrient per cell. Other types of structured population models would be like juvenile adults, right? You can think of a lot of different kinds. So before I take any questions on this formulation, let me show you just a little side tour as to where this comes from. Because this Q equation, it's kind of, to me, it was out of nowhere until Jeff Hausman explained it to me. He's like, it's just the quotient rule. I'm like, what? So, oh wait, well, we'll get into that in just a sec. First, this is just graphing. So this mu function, the growth as a function of the quota, this is a typical function we use from Michael Drope, I think. So it's this saturating function. There's a minimum quota where growth is 0, and then growth increases asymptotically as your nutrient quota increases. So we can interpret this as the minimum quotas, that's the nutrient that's like bound up in like actual functional machinery, and everything else is stored nutrient on the pile waiting to be assembled. And then we got this parameter mu infinity, which is not exactly mu max. Mu infinity is the growth rate you would have if your quota was infinite, but your quota never becomes infinite, so it's not exactly mu max, but it's kind of like mu max. Okay. This is the Droop function. He determined this experimentally. All right, so where's this weird quota equation come from? Here's a different way we could write the same model. So here we're gonna, instead of talking about Q, the nutrient per cell, let's just talk about two extensive variables, N, the total number of cells, and S, the total amount of nutrient stored like in the population. Okay, so I call them extensive variables because if we were to like double the population, we would double N and we would double S. So S is just nutrients in the population. So now this is a little bit easier to understand. So nutrients increase by uptake, which is now proportional to the size of the population. And then stored nutrients also die when an individual dies. Okay. And the equation for the N, the population size, is the same. It's still a function of the quota Q, which is now defined as stored nutrients S divided by N. Okay. So now we want to slip between this formulation and the one we showed before with an equation for Q. Well, this is how we do it. So Q is what we can call an intensive variable because nutrients per cell. So if we were to double the population, we would double N, but Q would just stay the same. All right, so yeah, it is literally just the quotient rule. So the dynamics of Q, we apply the quotient rule like here, plug in these equations from the left. These terms in red the mortality terms from the two sides, the numerator and the denominator, they cancel. Beautiful. Those red terms cancel. And all we're left with is this equation for the dynamics of the nutrients per cell, which is uptake minus dilution by growth. So anyway, this isn't like the most important thing in the world, but I just thought I'd put out there because it was nice when I finally saw where this came from. And people use these two formulations. You can go back and forth. Tomas a pro at that. Okay, all right. So enough about deriving this model. Wait, any questions on this? All right, well, I'll just tell you a few things about it. So we can simulate it. This is an example of simulation. We're gonna start out in our chemostat with nutrients high at the nutrient input concentration. That's green. Yellow here is the abundance, so we're starting out with just like a few cells. And then the interesting one here is the quota Q in blue. So you see that at the outset, the quota, we started at Q min, and they're taking up nutrients, but growth, and they're growing really fast. So their growth rate's high. So the quota kind of equilibrates for a while during the exponential growth phase. But then as nutrients are depleted, then the quota comes back down and the population approaches its carrying capacity. So this is what the dynamics look like. We want to understand this, particularly this two phases, the first phase, exponential growth phase, last phase at equilibrium. So here's a cool trick. So how do you sort out the quota during that exponential growth phase? And how do you sort out the exponential growth rate in this kind of structured population? So what we can use is this thing called a quasi-steady state approximation. We'll assume that the quota, nutrient quota, goes to equilibrium fast, which we saw in the simulations. And so we're going to take this quota equation, set it approximately equal to 0. We can rearrange it, solve for this Q twiddle, that stands for quasi-steady state, that twiddle. And so we see that the quasi-steady state quota is the minimum quota plus the uptake rate divided by the growth rate. So that makes sense. It's kind of like there's this war between nutrient uptake and dilution by growth. And then we can take this and plug it back into the population equation and get essentially the growth rate as a function of the nutrient concentration. So that's how we can understand this phase one. We could basically solve that expression is telling us what this quota value is and also the exponential growth rate that we would observe in an experiment. Also, we can, at the other end, we can just let this go to equilibrium, set everything equal to 0. What we end up with is a few things. So we get an expression for R star, of course, equilibrium nutrient concentration. We also get an equilibrium quota. And finally, we get the equilibrium abundance. What can I point out about this? The R star expression is still the measure of competitive ability in a constant environment. And so if you want to know who's the best competitor, just plug in all these parameter values and you'll see. But then everything else is kind of like before. So nothing else really changes a heck of a lot. And Burmaster showed back in 1979 that at equilibrium, this variable quota model is equivalent to the Monod model. So in some sense, at equilibrium, it doesn't matter. So why are we even bothering with this? A few reasons. First of all, for non-equilibrium conditions, it probably does matter. The sort of decoupling of uptake and growth. Second of all, experimentally, you can actually measure uptake rates and quotas and stuff. So this is, in some cases, a nicer model to parameterize. And finally, you might be interested in questions about stoichiometry, where the Q is like what you're after. So you want the Q to vary, not just like be fixed. All right, so I've got just a few quick things to wrap up here, and then we can get into our paper discussion. Anyways, this is just meant to be like a warm-up because it'll be explained in the paper as well for next week. The other thing, the reason I got interested in this is because there was a lot of experimental work done in the 70s with chemostats. And there was two particular types of experiments that are kind of orthogonal to each other. So one was done by Goldman. He would vary the dilution rate or the growth rate in a chemostat, and Ree would vary the supply of resources here, nitrogen to phosphorus. And so what Goldman found is in a chemostat, this is the dilution rate of the chemostat, which at equilibrium is the same as the growth rate. And here they were fed low N to P, medium N to P ratio of 5. And what you see is that like at low dilution rates, you match what you're fed. But at high dilution rates, this comes up and approaches this 15. Under phosphorus limitation, it's the opposite, right? The algae N to P would be more or less what they're being fed, and then it comes down to approximately the same 15. And Ree instead varied the N to P ratio at a fixed dilution rate and showed that there was basically, almost a 1-to-1 correspondence. So in this case, whatever you feed them, they take it all up, and that's what they are. So we wanted to understand these results. So we just took we just took all the off-the-shelf components and put them together in this model. So it's all the droop functions, it's 2 individual nutrient droop functions connected by Liebig's law of the minimum, Michaelis-Menten uptake, and that's in a chemostat. So that's the model. This is just showing some of the main results. Is basically it recapitulates those same patterns. So where you vary the dilution rate, you see the ratio of the quotas, which if you took the cells and like ground them up and like measure their nitrogen phosphorus ratio in the lab, that would be the N to P ratio. At low dilution rates, they match what they're being fed. At high dilution rates, they approach some sort of optimal N to P ratio, which is the ratio of their minimum quotas, the ratio that they actually require the nutrients in their biomass. And similar for the re-experiments, as long as you don't do it at too high of a dilution rate. All right, so anyway, that's just a like introduction to the droop model, variable stores, and so on. Any quick questions about that? So I want to save time to discuss the papers too. But we could take a few minutes. All right, well, I'll send these slides around too. So now we can get into paper discussion mode. Great. I'm glad I flipped the order like this. So here's the main paper. There were a couple add-ons, but they were kind of more limited in scope. And they tested the predictions of Tillman's resource ratio theory. Here's 7 predictions that they extracted, and they looked at papers that studied this since 1980, 1982. And here's the stats. This is shocking. 1,333 articles cite Tillman 80, 82. 85% of those is just like competition, parentheses, Tillman, very passing reference. 7.2%, good 7.2% extending the theory. But only 5% like actually decided to test the model somehow. And shocking one, number 2, of the ones that went to test it, they decided that like, I don't know, like 2/3 of them had like bad experimental design and couldn't actually test anything. And of the ones that did, the majority of the tests were done in microcosms in the lab, and only 4 field experiments testing resource competition theory. That's kind of shocking. Here's the table of support for the different predictions. Kind of a mixed bag, but I guess it's okay. And this is just showing the bias and the type of systems that people like to use. It's mostly like phytoplankton, which the theory kind of like grew up on. So that's the summary of the article for y'all, that if you didn't have a chance to read it. So here's some discussion questions I'll put in the chat as well. This is just some stuff I thought you could talk about in breakout rooms. And so what's that? So we'll break out in breakout rooms. You can discuss these questions and then whatever else you want to discuss about these papers. And we'll come back in say 17 minutes, and that'll give us 10 minutes to like compare notes. And so when you're in your rooms, assign someone to take notes or someone who's going to be willing to speak up. That's always key. So I'll start those breakout rooms then. I'll make 4. All right, so see you in a bit.